Hi everyone. Today we're going to talk about reproduction. It's fundamental to all of the aspects that we're going to cover in this course, ranging from ecology to evolution to organismal biology. One of the fundamental characteristics of life is that organisms need to be able to reproduce. So we're going to run the gamut today from looking at how single-celled organisms reproduce, and we are going to contrast the processes of cell division in prokaryotes and eukaryotes. We're going to make a distinction between what happens in mitosis and meiosis. We're going to talk about this process of fertilization because we are going to define sexual reproduction as a process where sperm and egg have come together, and that process is fertilization. So we'll look in more detail at what happens in that process. It turns out that major groups of eukaryotes, the plants, animals, and fungi, go through their entire life cycle using the processes of meiosis and fertilization differently from one another. So we'll look at what happens in those three groups and how it's different. And then we're going to look at differences between asexual reproduction, some kind of process that makes new individuals that doesn't involve sperm and egg, versus sexual reproduction, which is the kind that involves sperm and egg. And we'll look at the names of the structures that we use for those processes. So when we're talking about prokaryotes, these are very simple, fairly small cells. Going about doing reproduction is pretty easy for them. Cell division in prokaryotes is called binary fission. That just means splitting in two. It's a fairly simple, straightforward process. It can happen quite fast, only 20 minutes. Prokaryotes have no nuclear envelope, and they only have one chromosome. So keeping track of all of their DNA copies is not very hard. Right, so they're going to copy their DNA just like a eukaryotic cell does, and then they're just going to split in two. And like in eukaryotes, we're going to call those two cells where there was one, we're going to call them daughter cells. So the original cell doesn't exactly exist anymore. It has turned into two new cells, and that is going to be true of the process of cell division for both prokaryotes and for single-celled eukaryotes. All right, so we had one cell, now we have two. We can't find the original one anywhere. It's gone. It was the mother cell. Now there are two daughter cells that each contain elements that were from the original cell. If you are a single-celled organism, going through cell division is reproduction. And it is considered asexual reproduction because there was no fusion of sperm and egg. There was just a division of the cell, and the two daughter cells have exactly the same genetic information as the parent cell had, as long as nothing went wrong in that copying process. When we look at eukaryotic cells, they go through their life, and when they are going to divide or not is determined by a series of events that we call the cell cycle. So a normal healthy cell is going to have its cell cycle regulated, that is deciding when it's going to go through cell division based on processes that are happening inside the cell, how healthy it is, and also its environment. Does it have enough resources? And depending on the organism, what are the triggers for cell division? First, the cell has to get ready to go through division. And so we're going to divide the whole cell cycle, its entire journey from when it was first formed as a daughter cell. It's going to have to rest and grow for a while. So all of the period of the cell cycle that is not having to do with cell division, we're going to call interphase, which means in between, right? So it's everything that's going on when the cell is not going through cell division. So the most cells are going to be going through the process of mitosis. Meiosis is special. It only happens in certain body organs at certain times, and its only result, its only reason for being is to produce sperm and egg or gametes. So meiosis, M-E-I-O-S-I-S, is very specific to sexual reproduction, whereas mitosis is for everything else. If you're a eukaryotic organism, mitosis is going to be used for cell growth, body growth. It's going to be used for asexual reproduction if you're a single-celled organism. It's going to be used to repair damaged structures and any kind of cells that need to be replaced during the lifespan, of the cell will be done by mitosis. So a quick and easy way to remember this is for persons with ovaries, this works great. Meiosis happens in my ovaries and mitosis happens in my toes. Those of you with testes, you can, sympathetically remember that the ovary is the sexual reproductive organ. All right, meiosis in ovaries, mitosis in toes. Okay, so we have a few things we need to do in order to get ready to do cell division of any kind. One of them is the cell needs to double basically all of its internal contents and carry on with its normal cell life processes. It's going to do that for a little while. So interphase is broken down into 3 parts. We call the first part G1, the second part S, the third part G2. G1 and G2 are sort of resting, growth parts of the cell cycle. We aren't going to talk about what happens in those parts of the cell cycle. That's a Bio 230 topic. But we do need to remark upon the fact that at some point during its cell cycle, the cell is going to do DNA synthesis. It needs to have 2 complete copies of all of its DNA ready to go if it's going to divide, because it needs to put one complete copy of all of its DNA into each daughter cell. So DNA duplication, replication, or synthesis is going to happen in that S phase of the cell cycle. Notice that it happens a little while ahead of when mitosis actually happens. So the cell wants to make sure all of its DNA is copied and ready to go, and then it's going to busy itself about some other things for a while. And when it's ready to start, it's going to do this process called mitosis. Mitosis refers to moving the chromosomal material into 2 new nuclei, whereas cytokinesis refers to splitting the cytoplasm, right? You can remember that because cyto is the beginning of the word and kinesis means movement or motion. So cytokinesis is the part where the cytoplasm and the cell membrane actually rearranges in order to split or separate the 2 new cells from one another. So mitosis has to do with what's happening with the chromosomes and the nuclei, whereas cytokinesis has to do with the cytoplasm and the cell membrane. So cytokinesis is actually going to begin once the cell is partway through mitosis, and then those processes are going to kind of go parallel for a little while. At the end of cytokinesis, you have 2 daughter cells, and each one of them will start its own cell cycle. And if it's the right circumstances, then they may go on to do mitosis again. All right, so the DNA material in eukaryotes is going to be made of more than 1 molecule of DNA. Each separate molecule of DNA is called a chromosome. Organisms have different numbers of chromosomes depending on their evolutionary history. Some species have 10, some species have 4, humans have 23. So you can think of the DNA molecule as being so big, it has to get broken up into different parts, right? So a book has different chapters, or blueprints for a building has many pages. So what genes are on which chromosome is kind of arbitrary, but for many organisms, we now know which genes are on which chromosome, so we can talk about them as having specific information. So in a normal animal cell, during its normal activities, the DNA material is going to be spread out across the nucleus. But when it's getting ready to do mitosis, it needs to be able to keep track of all of the chromosomes and not get them tangled up with one another. And so what's going to happen is that DNA material is going to go from being all spaghetti noodled and packaged up and coiled into very tight bundles. And at that time, we really are seeing each chromosome separately from the others. And so when the cell is getting ready to go through mitosis is a time when we can actually visualize the chromosomes as separate entities to one another in a light microscope image of the cell nucleus. All right, so each chromosome is going to be a single molecule of DNA. And then after the cell has gone through the S phase of the cell cycle, it will have duplicated each chromosome. So what we'll see in the nucleus is that we'll have each chromosome that the cell originally had and its exact copy, if everything went well, there were no mistakes. And that these two copied chromosomes, remember that in DNA synthesis, the DNA molecule unzips and then we get new material on each side. So the two chromosomes of DNA material that we have after replication are each half of old and half of new. They should be identical to one another. And what's going to happen is that these two copies, these two identical copies, are going to be fastened together with a structure called the centromere. So if we want to make sure that exactly one copy of each of these chromosomes goes into each daughter cell, we need to keep track of which one goes with which copy, right? And the way to do that, the cell does that, is by keeping them fastened together with the centromere. When the cell is ready and it has the cytoskeleton fastened to the poles where the new nuclei are going to be, and it has it fastened to each of those paired sister chromatids, then the centromere breaks and we're going to pull each one of those identical copies to the two new nuclei. All right, so not only are these called sister chromatids, they should be identical twin sister chromatids. All right, so when we're talking about mitosis versus meiosis and what's happening in the life cycles of plants, animals, and fungi, we need to have a very good understanding of how many sets or how many copies of sets of chromosomes we have in the nucleus. So we're going to talk about this a little bit more. We're not going to go through all the steps of mitosis and meiosis. You're going to go over that in your Bio 230 class. We're not going to talk about, prophase and metaphase and what's happening in those. We just want to talk about the copies of chromosomes and what happens to them. The fundamental concepts here have to do with the ideas of diploid versus haploid, and you need to really understand that to understand a lot of stuff in Bio 240. Right, so animals, like ourselves, when we are adults, our bodies are going to have in every nucleus of every cell two entire sets of our chromosome. So we're going to call that number of chromosomes we talked about earlier, that quantity, whether it's 10 chromosomes or 23 chromosomes, or in this example, 3 chromosomes, we're going to call that little n, right? So little n is how many different chromosome molecules do we have that makes up one complete set of our DNA. We are diploid as adults, animals are, so we're going to have 2 complete sets of those chromosomes. So this diagram is showing a cell that has gone through the S phase of the cell cycle already. It's getting ready to do. It could be mitosis or meiosis. It hasn't necessarily decided yet in this image. And we have one complete set of chromosomes that we got from our mom, from the egg, and we have one complete set of chromosomes that we got from the dad in the sperm, right? So the process of making gametes actually has a very important task of taking one complete set of all the chromosomes and packaging them into those daughter cells, so that when sperm and egg come together, we have taken one set of the chromosomes, added it together to the second set that comes from the other gamete, and we're back to our normal complement, which is two complete sets of the chromosomes that make up our genome. So in this simplified diagram, we can see that each of these sets of identical twin paired sister chromatids is in these little, little X-like formations. So they're fastened together with a centromere. So we have three different chromosomes, the long one, the medium one, and the short one. And we have one version of those chromosomes that we got from mom and one version that we got from dad. So if there's a gene for eye color on chromosome 1, the big one, right, we'll have one version of that gene that we got from our mom's DNA, and we'll have one version that we got from our dad's DNA. So when we think about our genetic information and we're looking at Mendelian genetics and we say, oh, we have two alleles, we could have a big A and a little a. The reason we have two alleles is because we have two sets of chromosomes that make up a normal complement in our cells. We got one from our mom and one from our dad. Now, what our actual eye color turns out to be depends on what protein those genes make and how the information from those two chromosomes interact with one another, right? That genetic information could be the same on both the chromosome you got from mom and the one you got from dad. You could have blue eyes, in which case your only choice is to end up being blue eyes. Or you could have two different versions of that gene, right? So we'll come back to that later on in the semester. But if you already know a little bit about Mendelian genetics, I want you to try and relate that to this idea of being diploid, that is having two complete sets of the genes that you need to make and run the body. All right, so what's going to happen in mitosis? Very simple. You're going to have all of these duplicated pairs of identical sister chromosomes. They're going to line up, and then each one of them is going to separate, okay? So when we talk about our complement of DNA in an animal adult cell as being 2N, we have 2 times however many chromosomes there are, right? So this organism has 3 different chromosomes. It's got 2 sets because it's got one from mom and one from dad. That means 2 times little n is going to be 6. Right now, if we count up all the chromatids here, though, then we're going to see that we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now, after a cell has duplicated its DNA, we've duplicated that 2n, right? So we've gone from having 6 distinct chromatids to having 12 chromatids. Now they're exact copies, but we still have to count them, right? So whenever we see a picture with these chromatids fastened together at their centromeres, then we know that we're seeing a cell in its 4n state. Now this is transient. It's only going to be until the cell goes through mitosis or meiosis. So we normally don't have organisms running around in a 4n state. Right, so mitosis, very simple. We're just going to split apart each of these chromatids. We're going to end up, we started with one 2n cell. It went through DNA synthesis to become a 4n cell. So we're going to split it apart and we're going to get 2n cells. Right? We've got to have conservation of chromosomes. Right? So each cell has to end up with the right number. Product of meiosis, we start with a diploid cell, we should end up with 2 diploid cells. All right, here's another way of looking at the entire genetic composition of a human. So when we say we have 23 chromosomes. We can actually take a picture of them. It's called a karyotype, that's with a K. And when we use particular dyes and stains, the different chromosomes will get these different colored striped bands. So between the size of the chromosomes, where the centromere is, whether it's at the top or the middle or the bottom, and where the colored bands are, people who do this for a living can identify which chromosome they're looking at. They can say, oh, well, that one's chromosome 12. All right, so traditionally they're numbered from largest to smallest, and we don't ever see them in the nucleus of the cell looking exactly like this. The way this picture is made is that cells that have gone through the S phase of the cell cycle are visualized using light microscopy after they've been stained. And lots of different cells, nuclei, have pictures taken of them. And then basically you cut out the best picture of each of the different chromosomes and assemble it together into a collection like this. So they don't lay like that in the nucleus already for us to look at them. So somebody has done a lot of work in order to make this nice picture. So in the human genome, we have a chromosome that is used mainly for determining sex. So we have an X or we have a Y chromosome. We have two, we have an X, we have a Y. We have a female individual if we have XX. We have a male individual if we have an XY. So when we're talking about what is a normal complement of our genetic information, that is exactly 2 copies of each of our chromosomes. It's possible if there is something that goes awry during mitosis or meiosis, that we get daughter cells that don't have exactly 2 copies of every chromosome. So when we have that happen in the germline, in the sex cells, that ends up going into the body and forming the body of the offspring. And certain chromosomes, we can have extra copies or we can have fewer copies, and it doesn't make any difference. Some chromosomes that if we have fewer copies or extra copies of, the embryo dies, it's unviable. You cannot, it just doesn't work to have that information missing or to have extra information. Some of them cause predictable sets of characteristics. So for example, if we have trisomy 21, that's a condition where we have certain kinds of symptoms. We call it Down syndrome. And it's relatively common, especially with older moms, for this to happen. All right. So we're going to call these 2 copies of the same chromosome, we're going to call those homologous pairs. So when we use the term homologous pairs, that means chromosome 13 that you got from your mom and chromosome 13 that you got from your dad. They're homologous to one another. That means they have the same list of genes on them in the same order, more or less, and that those are basically 2 copies. Now, there's a benefit of being diploid, and that is for some genes, if we have a mistake in one copy of that gene, we have a backup. And so that we may not have any negative consequence of having a problem with a gene, because the other copy of the chromosome that we have can provide us with the correct or normal or functional version of that gene. Oops. All right, so this is a quick slide showing the processes of mitosis and cytokinesis. They look a little bit different in plant and animal cells. Animal cells are at the top, plant cells are at the bottom. So plant cells have a cell wall and they basically stay in the same place. They're more like a room in your house. Animal cells are usually more of a rounded shape and they don't have a cell wall, so they have a cell membrane and they're much more squishable. This is a nice kind of visualization that can be done with fluorescent tagging. So we have different colors that are tagging different things, and we can see what happens in mitosis using this technique. So the cytoskeleton is stained in green and the chromosomes are stained in blue. So we can see that when the cell is busy being a cell in interphase, the chromosomes are spread out over the whole nucleus. They have not condensed. We don't see them separately. And the cytoskeleton is also distributed throughout the cell. Once mitosis starts, the spindle is going to form, the cytoskeleton that grabs on to the chromatids and it pulls them to the two new nuclei. And then we have this pinching in of the cell membrane to separate off the two cells from one another. In plant cells, it's a little harder to see, but basically what happens if you look in the middle of that middle cell to the left and then to the right, you can see that there's now a partition. So what the plant cell does is it moves the new nuclei to the two ends of the cell, and then it puts up new cell wall, and then it grows or it sort of stretches in length, right? So plant cells kind of start out by saying, hey, we need a new room. Let's just put up a partition in the middle. All right, now we have two rooms. They're each half the size that they were to begin with. So cytokinesis looks different in plant cells and animal cells. All right, in meiosis, we really need to understand the concept of those homologous pairs of chromosomes. So this example is showing if we only have a single chromosome in the entire genome, right? So little n here equals 1. We start off with a cell that had one copy of its one chromosome from mom and one from dad. It's going to start by going through cell replication or DNA replication. So it's going to go from having 2n to having 4n. You can see those pairs of sister chromatids after step 1 in the diagram. And what's going to happen in the first part of meiosis, right? So we have to get from 4n to 1n cells, right? Conserving our chromosome number. That means we're going to have to go through not just one cell division like we did in mitosis, but we're going to do 2 cell divisions. So meiosis is different than mitosis in several ways, one of which is that it takes one 2n cell to duplicates the DNAs until it has 4n, and then it divides twice and ends up with 4 1n cells, right? A 1n cell is called haploid, right? So a 2n cell is called diploid. And then the product of meiosis is going to be those gametes, all right? We have to have our gametes haploid because we want to fuse 2 of them together and return to the diploid condition, right? So another way to think about meiosis is that it takes diploid cells and it produces haploid cells, right? Those haploid cells are the gametes. So this process of meiosis would either be making sperm or it would be making eggs. So the first cell division of meiosis, we're going to just separate those homologous chromosomes. So chromosome that you got from your mom and the one you got from your dad, and each of their exact copies are going to separate from one another. Then in the second round of meiosis, the second cell division, basically the same thing that happened in mitosis is going to happen. The paired sister chromatids are going to separate. All right, so the centromere doesn't break until the second cell division in meiosis 2. All right, this is a bit more complicated if you have more than one chromosome. We're going to do an activity, a visual activity to practice thinking about what happens in mitosis and meiosis later on, right? For the purposes of this class, the reason I'm talking about it is not because I'm going to give you a test on all of the ins and outs and details of mitosis and meiosis, but because it's really, really important to understand the difference between these two processes, what they start with, what they end up with, and the concepts of haploid versus diploid. All right, so we're going to shift gears now and talk about this process called fertilization. So fertilization, we are going to define it as the fusion of sperm and egg. And what happens is that the nucleus of the sperm actually goes inside the cell membrane of the egg. All right, and so basically the cytoplasm of the egg, the cell membrane of the egg, the nucleus of the egg are all still there. What you get from the sperm is the nucleus, and its nucleus moves inside the egg. The sperm nucleus is haploid, the egg nucleus is haploid. When they fuse together, they become a diploid cell, and they are no longer sperm and egg. They are a fertilized egg, which is called a zygote. All right, so the process of fertilization is basically going to be the fusion of sperm and egg to form a zygote. That is our goal, to make this zygote happen. So we have some different structures that exist on the egg. We have some different structures that exist on the sperm. And the functions of these structures have to do with the sperm and the egg recognizing each other. The sperm needs to be able to fasten to the egg. It needs to be able to cause the cell membrane of the egg to change in a way that will let it put its nucleus inside, because most of the time, two cells, they do not try and share cytoplasm with one another. We also have some structures that have the function to prevent more than one sperm from fertilizing the egg, right? So if more than one sperm nucleus gets inside the egg, we'll have the wrong number of total chromosomes. If one extra sperm gets in, we'll be triploid. If two extra sperm get in, we'd be tetraploid, and so on. We're just generally going to call too many sperm polyspermy. Right? We want to prevent polyspermy, which would be more than one sperm fertilizing the egg. So there's actually two mechanisms that help prevent that from happening, right? This is a very, very important thing to prevent from happening, right? So there's two mechanisms. So we're going to pause in our conversation about what happens during fertilization to mention where these blocks to polyspermy are. So we have something that happens that is going to prevent more sperm from fusing with the egg. And those are called blocks to polyspermy. All right, so we have some stuff that's happening on the sperm's side of the picture. And so we have a sperm coming in here, and we have this structure here called the acrosome. So the stuff the sperm is doing is generally going to be referred to as the acrosomal reaction. And we have these structures that are inside the egg that are called cortical granules. You notice those over here. And so the stuff that the egg is doing as part of this process is called the cortical reaction. So the things the sperm is doing, well, first of all, it's going to swim towards the egg. It actually is capable of chemosensory of chemosensing. So it can smell, right? So you should think of the sperm as being able to detect a gradient of molecules that the egg is releasing, sort of like a moth sitting there releasing pheromones, attracting male moths. The egg cell is doing the same thing. The sperm can smell it. It will swim towards the egg. Once it gets there, in order to get the cell membranes of the sperm and the egg together, the egg is protected by an outer layer that's a gelatinous material, and it's called the jelly coat, the jelly coating. And so the sperm has to break down this coating and in order to get close enough to the membrane of the egg. So the first thing that the sperm does is it releases some enzymes. They're hydrolytic enzymes. That just means that they're going to break apart biological macromolecule bonds. So they're going to break down the structure of the jelly coat. So what's happening in this process, we're reading on this diagram from left to right. So the first thing is the sperm swims up. The second thing is that the structure of the sperm includes this little vesicle that's full of these enzymes. It's going to do exocytosis so that it releases those enzymes onto the surface of the egg, which are going to digest and break down the jelly coating. Once that happens, it's going to use the cytoskeleton to build an extension out the front of the sperm, right? So this structure is called the acrosomal process, and a process is something that sticks out. So it's basically like just gonna build this structure out of proteins that sticks out of the front of the sperm. And it has a particular molecular shape and structure. So little structures that are sticking out of the sperm, they have to line up with, match exactly, and bind to structures that are on the surface of the egg cell membrane. So these little structures that this diagram shows, they're like little Ys that are purple. And you can see that these balls, little balls from the acrosomal process have landed into these structures. So if you think back to how enzymes work and how the enzyme and the substrate have to fit together like a lock and key, the same thing happens with the tip of the acrosomal process and these proteins that are on the surface of the egg membrane, right? The proteins on the surface of the egg membrane are called sperm binding receptors, right? A receptor is going to be any protein that's sticking up from the surface of the cell that's there for something to bind to. Now, these receptors are also a way that, say, viruses bind to our cells, right? So there's lots of different receptors that are sticking up on different types of cells. So these happen to be the sperm binding receptors, right? The sperm binding receptors are found on the surface of the egg because the sperm wants to bind to them, okay? All right, so why do we want to have a lock and key fit between the sperm binding receptors and the acrosomal process? Well, because we want to make sure that the sperm from the right species is matching to the egg, and that if fertilization happens, that we're going to have a resulting individual that's going to be viable, right? So this is going to be a mechanism that makes sure that we're having that we're keeping our species intact. So we'll talk about this a little more when we talk about species in a few more lectures, but the sperm binding receptors have to match up together. Now, very closely related species may have similar enough receptors that they can fertilize, but if you have distantly related organisms and you put the sperm and the egg together in a dish, they will not bind and fertilization will not happen. All right, so once the sperm has bound to the receptor, then the cell membranes of the sperm and the egg are going to merge together, right? They're going to fuse. It's kind of like the sperm is doing exocytosis of its whole nucleus. And in order for that to happen, it has to fuse cell membranes with the egg cell. Right, this is a very, very special event. This fusion of cell membranes hardly ever happens in living things. And so this sperm and egg cell membrane fusion is a very special and important component of this event. All right, now what happens when their membranes fuse is that there is sort of an electric shock that happens to the whole egg cell membrane. And it triggers sort of a change in polarity of the cell membrane. And it is going to temporarily cause a change in the cell membrane that's going to prevent another sperm from fusing its cell membrane. It happens quickly. It happens right after the sperm binds. But then the egg cell is actually going to reestablish homeostasis after that, so it doesn't last very long. So this one is called the fast block to polyspermy. That is the depolarization of the egg cell membrane that occurs when the sperm and egg cell plasma membranes fuse, right? It is happens rapidly, but it disappears, right? So if we didn't have a permanent block to polyspermy that happens afterwards, then we would still be vulnerable to polyspermy. So the next thing that happens actually induces that permanent block to polyspermy. So if you want to prevent any more sperm from binding, well, you can cut off all of the rest of the sperm binding receptors and no more sperm can bind, okay? So we're going to switch gears now to things that the egg is doing. So we finished talking about the acrosome reaction. Now we're going to switch gears to the cortical reaction. The cortical reaction is where the contents of these cortical granules are released outside the cell membrane of the egg. So these cortical granules are just vesicles that are lined up, ready to go on the inside of the egg cell membrane. And they release a substance, a mixture of substances that we don't totally know what they are, but basically they cause a rush of water into the space between the egg cell membrane and this structure called the vitelline layer. So this vitelline layer is shown in dark purple, and it's made of sort of sturdy but stretchy molecules. So what happens when the cortical granules release their material into this space is water rushes in and it stretches the vitelline layer. And as it stretches, it lifts away from the surface of the cell membrane and it just rips off all the rest of those sperm binding receptors. So we can see this happening when we're looking at an egg that's getting fertilized under the microscope. And it actually you see it lift off and then sort of form all the way around the egg. It looks like almost like an egg white when it's sitting in a pan. You've just cracked it in a pan with the egg yolk. It's very clear, but a distinct line. So now we say that this egg has been fertilized, we can see it, and that structure we now call the fertilization envelope once it has formed, right? So the formation of the fertilization envelope is the second block to polyspermy, and it's called the slow block because it happens, it takes more time for it to happen, right? But it's permanent. All right, so there's a lot of structures here. You need to know the names of those structures and their functions. What do they do? Okay, so where they are, how they relate to one another, those are the kinds of things to think of and the order that they go in. All right, so what happens after fertilization? Well, if it's a normal cell, we're going to get the zygote. So we have the sperm and egg fused together. In these pictures, you can see the fertilization envelope. It's very dim in the first image, but in image B, you can see it as that very light halo that's surrounding those two cells. So what's going to happen to the zygote is that we want to build a multi-celled organism. So we start with a single cell. We're going to have to start dividing to make a multi-celled organism. So that usually happens right away. Sometimes within an hour, we get the first cell division. So this set of cell divisions that happens right after fertilization is called cleavage. Egg cells are really, really big. So what happens is that these cells start to divide really fast and they don't grow in the intervening time period. So the cell cycle for cleavage in an early embryo, so once we have a zygote and it starts dividing, we usually call it an embryo. We have a 2-cell embryo and then a 4-cell embryo and then an 8-cell embryo and so on and so forth. And we'll come back to this later in lab for animals. And we'll eventually form in animals, we'll eventually form an embryo that has a particular structure shaped like a hollow ball. And the cells are very, very tiny compared to the zygote. So if you look at the blastula on the lower right-hand side, that's the basic body of an animal that's just getting started to turn into the body of an animal. So we'll talk about later on how this changes into all the wide variety of different animal bodies. But basically, we all start out as a single cell, a fertilized egg. It rapidly divides until it forms a hollow ball shape called a blastula. And then animal types and different phyla of animals start looking different from one another after that. All right, so we've been talking about animals so far. So if we want to draw a diagram of what happens to animals during their life cycle in terms of sexual reproduction, right? So we've got fertilization on this diagram. So that means we're talking about sexual reproduction only. We start out this cycle at the top, if you want to, with gametes. They're haploid. That's being represented by 1N. And if we have 1N, we don't always write the 1. So we have two gametes that are coming together for fertilization. That's our definition of fertilization. They form a 2N zygote. Now, for multicelled organisms, we're going to immediately start going through cell division. And that cell division is going to be a mitosis cell division. It's not going to be meiosis, right? So we're going to grow from single-celled to multicelled. The zygote is diploid, so the multicelled organism is also going to be diploid. Now, somewhere in its body is going to be a special place, a sexual reproductive organ, where meiosis is going to happen, and meiosis is going to produce gametes to make the children of this organism. Okay, so this diagram isn't trying to show that an organism is going to fertilize itself, right? It's just trying to show that a gamete has to fuse with another one, then that's going to grow into a multi-celled individual. All right, so our life cycles are all going to have, these are sexual life cycles, they're all going to have fertilization and meiosis in them. All right, this should seem pretty straightforward and normal because this is animals, this is what we do, we're familiar with it, it makes sense. All right. Now here's fungi, the life cycle of fungi. So think about a mushroom. It turns out they have turned this life cycle on its end. So notice that we still have meiosis and fertilization. So fungi do sexual reproduction. But look, when they fuse two gametes together into fertilization, it forms a zygote. But instead of growing by mitosis into a multi-celled individual, it immediately goes through meiosis. So instead of the gamete being transient in the life cycle of fungi, like it is in animals, the zygote is transient. So we do not, in the fungal life cycle, we don't ever find a multi-celled individual that's diploid. All right. However, when they go through meiosis, they make cells. Now, these cells are not gametes. Okay. Gametes we're going to define as cells that fuse together in the process of fertilization. What happens to these cells is that they go through mitosis and become a multi-celled haploid organism. So the mushroom that you eat on your pizza, if you could do a karyotype of its genome, you would see that it is haploid, not diploid. All right. So because it's biology, we are going to have a different name for something that's different. So these are not gametes. Over here, these are spores. All right, and we're going to define in the context of these sexual reproduction diagrams that the product of meiosis, if it goes on to divide and become a multi-celled organism, we're going to call it a spore. Right? The product of meiosis, if it fuses together to become a zygote, it's going to be called a gamete. Right? So there's two different types of product of meiosis depending on the life cycle. All right, so animals only have a diploid multi-celled individual. Fungi only have a haploid multi-celled individual, whereas plants, they couldn't get enough of everything, and they have both. All right, so basically we see the bottom half of the animal life cycle here fused together with the top half of the fungi life cycle. They do it all, right? Well, what does this mean? This means that in the life cycle of plants, there's two different kinds of multicelled individuals, ones that are haploid and ones that are diploid. And in most of the plants that we see around us every day, these individuals look very different from one another. Early in the evolution of plants, however, if we look into the algae that live in the ocean, which came from the ancestor of land plants, that there's many kinds of algae that have these diploid and haploid multicelled individuals. They actually look identical to one another. You can't tell them apart unless you do a karyotype of them, or they start to grow reproductive structures. All right, so because we have these two different kinds of individuals, haploid ones and diploid ones, they have different names. And we talk about this plant life cycle as being an alternation of generations. A parent is going to be diploid, is going to have an offspring that is haploid, and then that haploid individual is going to have an offspring that is diploid. So every generation, from one parent to the offspring, is a generation. Every generation we switch from being one kind of individual to the other kind of individual. This is really weird. We don't do it. Animals don't do it, right? So the haploid multicelled individual is going to be the one that makes the gametes, so it's called the gametophyte. The diploid multicelled individual is going to make the spores, so it's called the sporophyte. And we will talk about this a lot more as we go through, mostly in lab. We're going to come back to this when we talk about animals and plants and fungi. All right, so we have organisms that are going about doing sexual reproduction. We just are going to wrap up by talking about the body structures and sort of different ways that organisms are going to go about doing sexual reproduction. We're going to talk a little bit more about asexual reproduction in multicelled organisms. Humans don't do asexual reproduction, so it's a little weird to think about it, and I'll give you some more examples. All right. So we can have sexual reproduction in animals and plants where we find both male and female reproductive organs in the same individual. Humans aren't like this. We have separate males and females, right? So we have different bodies that have each of those male and female reproductive organs in them. When we have an organism where the individual has both male and female reproductive organs, we call it monoecious, right? Monoecious, has both male and female reproductive organs. Sometimes organisms that have this can be self-fertile, right? So sometimes they may be able to put sperm from their male part into their, vagina or reproductive opening of their female part. Now, sexual reproduction, one of its benefits is that it brings together genetic information from different individuals. We mix up that genetic information and we get new combinations that may be beneficial, right? Especially if the environment changes or the individual moves to a new environment, right? So where we have sexual reproduction, usually self-fertilization is a last choice. The organism isn't going to want to do that. It's going to try and outcross or mate with a different individual if it can. All right. So often these organisms have a mechanism to prevent self-fertilization as well. A lot of plants are going to be monoecious. When we look at the structure of a flower, if we find both pistils and stamens inside the flower, that plant is monoecious. It has both male and female reproductive organs in the same body part. Some plants have separate males and females, right? We'll find flowers on one plant, the individual is male, it only has the stamens, and a different individual plant somewhere else has the female parts. If we have separate males and females, all right, so we had monoecious, all right, monoecious as our word here. If we have separate males and females, separate sexes, it's not monoecious, it's dioecious. So you're going to take that mono off the front of this word and you're going to make it a di. Dioecious. This is an I. Sorry, I had a glitch. Dioecious. Having separate sexes. Two houses. Right. Now, when we look at every kind of animal with sexual reproduction, it has essentially the same body parts. There's going to be, in the male, the place where meiosis happens, all right, the gonad or the male reproductive organ is going to be called the testes. There's going to be some tubing that connects the testes to some structure that will allow it to get the sperm to the outside world, and we'll call that structure the penis. Right, so the penis is going to be some kind of tubing that may or may not project to the outside of the body of the male, and its purpose is to get the sperm somewhere near the egg. The female body organ that is going to make the eggs is the ovary. So meiosis is going to happen in the ovary in the female, and there's going to be some kind of tubing that's going to connect to an opening to the outside world. All right, we're going to call this the vagina, most organisms. These other glands sort of provide stuff that is going to help successfully move the sperm or the egg to the place it needs to go, provide it with nutrients, modify the environment so that it is accommodating for the physiological needs of the egg and sperm. So we have male, we have female. Honeybee, once she has mated, she has this structure called a spermatheca. She stores sperm there. She may mate with multiple males and then fertilize eggs for the rest of her life with the sperm from those different males. Some species can choose which male they fertilize the eggs with. All kinds of really cool and interesting biology about mating and males and how males are going to try and get their sperm to be the one that fertilizes the egg, and how females are going to have strategies so that they can maintain their choice. All right. So when we're talking about organisms that have a penis and a vagina, that is a way for the sperm to get into the body of the female. So the mating is going to happen. The penis will introduce the sperm into the vagina. It will be close to where the eggs are. So if that's happening, we are going to have internal fertilization. So internal fertilization. That's internal to the body of the female. Right, that doesn't necessarily mean that the rest of the development of the eggs is going to take place inside the female. She could lay the eggs after that. Okay. Now, the opposite of this is external fertilization. So external fertilization can only happen in aquatic environments. If we just drop sperm and eggs onto the ground out in the dry air, they will die. So when we look at marine organisms or aquatic organisms, we're going to see a lot of external fertilization. Sometimes the eggs and the sperm, like corals, are just going to all release their eggs and sperm and hope for the best, and they're going to mix in the water over the reef. Sometimes, like frogs, the male will grab onto the female, and so he knows which female he's fertilizing the eggs for, and the female knows which male he's fertilizing her eggs. She'll lay her eggs, she'll push them outside of her body, and the male will release his sperm over them so that they get fertilized. So that's external. There are pros and cons of internal versus external reproduction, but if you are in a terrestrial or on land environment, the organisms need to do internal fertilization for the most part, because the external environment is not conducive to the life of sperm, eggs, or tiny early embryos. So there's a weird exception. Sometimes the male is going to, both in aquatic and terrestrial environments, he's actually going to make kind of a package of his sperm in a protective coating. that's going to be called a spermatophore, and he'll be able to hand it to a female, and she can insert it into her reproductive structure. So it's not very romantic, but that's done in some spiders, it's done in octopus, and a variety of different organisms. When we have an act where internal fertilization is happening, the penis is inserted into the vagina, we usually call that sexual intercourse in human lingo, but we would generally call that copulation. If you think of a couple being together, then copulation is the insertion of the penis into the vagina for the purpose of putting the sperm inside the body of the female in a friendly environment close to where the eggs are so that fertilization can happen. In a human male, we basically have the same structures. The only one I'm going to ask you to know are the testes, which is where the sperm are made. Then we have some tubing that's going to connect to the outside world, and the penis is the copulatory structure. So mammals have internal fertilization that requires that there is some kind of penis structure and vagina structure that match together in a way they fit in the same size and shape, in a way that bodies can move to get them close to one another. And so there's a lot on this slide, but you don't need to know anything but testes and penis. Same thing for female mammalian reproductive anatomy. The ovaries are the place where the eggs are going to be made. The oviduct is going to take those. It's going to pass through. The egg's going to get to the uterus. The vagina is the flexible tubing that connects to the outside world where the penis will be placed in order to get the sperm into location to swim up through the uterus, find the egg, and fertilize it. Now, the uterus is going to be a place where the developing embryo in mammals is going to be nurtured, kept, raised carefully internally, and then the baby will be born when it's ready to be in the outside environment. So lots of different organisms, whether they grow their offspring internally or not, may still have some kinds of parental care, right? So we can have, as organisms are growing from very early embryos, we can have internal development, like we were just talking about in mammals, where the embryo stays inside the body of the mother. Or we can have external development. So not just in aquatic environments, but several groups of terrestrial organisms have evolved ways to have external development of the embryo in a safe, protected structure that isn't going to dry out, and it has all the nutrients that the embryo needs to continue growing until it's ready to hatch or germinate out of its structure. So we call those eggs and seeds. We have reptiles and birds lay an egg that has an embryo developing inside of it. So they're going to have development of the offspring external to the mother. There may or may not be parental care, but there's going to be a little embryo developing inside the egg. It's kind of like a little spaceship that has everything inside of it that that embryo needs. This is in contrast to fish and amphibians. They're basically going to be just in kind of a gelatinous coating that has to be laid in water. All right, so that's an amphibian or a fish egg. And the reptile or bird egg is called amniotic because it has an amnion, which is the membrane that surrounds the embryo. The amphibian or fish egg is called anamniotic. It doesn't have an amnion, all right? It doesn't have a shell either, right? So those are going to be like a fish egg. Now, the equivalent for plants of an amniotic egg is a thing called a seed. So if you buy a packet of seeds from the store, you plant them in the ground, they're basically the equivalent of a chicken egg. They have got a little plant embryo inside. It's got enough nutrients to keep it going while it continues to develop and grows into a new baby plant. All right, so we have external development in both land plants and in reptiles and birds. Where we have internal development in mammals only, we have a very complicated structure called the placenta that allows the transfer of nutrients and oxygen to the embryo and allows transport of wastes from the embryo away to be processed by the body of the mother. So there's a really, really interesting structures of the placenta. We don't have time to get into it in very much detail here, but I'll answer questions about it when we have a chance to talk. All right, our last topic is asexual reproduction in multicelled organisms. This is really common in plants, although it happens in animals as well. So we're going to start talking about plants. Asexual reproduction in plants is going to be growth vegetatively or from part of the body of the plant. And it's any kind of reproduction in plants that doesn't have a seed, right? So most flowering plants and other kinds of land plants have a process of fertilization where the sperm finds the egg and then an embryo grows, but this is not that, all right? If we have plants with bulbs underground, like garlics, those are going to grow these little separate partitions, and then they're going to break apart from one another, and each one can become a new separate plant. So if you want to grow lots of garlic, you can buy one head of garlic, break it apart into all those different parts, plant them in your garden, and if you're lucky, each one grows separately into a new plant. They should be genetically identical to one another. We can just cut a piece of a plant off, and under the right circumstances, it will grow roots and it will start to grow again as a new individual. Right? This would be like if you just, chopped your hand off, stuck it in a jar, and it grew a whole new you from the hand. Right? So this is a pretty neat thing that many plants can do. They can also sprout up these little babies from around their base, from the roots. Eventually, these can break off. If an animal comes by and just disturbs them, they'll break off and they can get transported away. Sometimes it'll grow a long shoot off that'll have a new baby plant at the end of it that'll start to grow roots, and it can grow as a new individual. Right, so for multi-celled organisms, asexual reproduction can be a great thing to do. Your body breaks apart, and instead of dying, you grow into several new individuals. Seems like a win, right? And the only thing that is unique about asexual reproduction in plants and animals is that it does not start with a single cell. It does not start with sperm and egg coming together. Right? So any other kind of process that results in a new individual being formed is going to be asexual reproduction. And in asexual reproduction, the offspring, the new individual, should be genetically identical to the parent. All right, in animals, we have some similar kinds of things that happen. We can have a new baby sort of just grow off the side of an adult. We can call that budding, which is much like what plants do. We can have some different kinds of organisms. If you break off a piece, you break a starfish in half, each side will regrow and you'll end up with two. So instead of dying, you end up with two. And some completely mature adult individuals can just split into two, right? That doesn't happen with complex organisms, but with animals that are very simple, like this anemone, they'll do that. So that's called fission. A really interesting example that is still asexual reproduction is parthenogenesis. Parthenogenesis is where a female individual is able to have female offspring that come from an egg that starts to develop and grow without being fertilized. So it's a strange intermediate between sexual and asexual reproduction. However, because no sperm is involved, it's considered asexual reproduction, and the offspring are genetically identical to the mom. So at some point, the DNA has to be duplicated depending on the organism. It will happen in different ways. Right, so after the babies are born, different types of organisms may have some kind of parental care, right? And this cartoon is making, this Far Side cartoon is making fun of a of a chicken who didn't know the fact of life, and she was putting her eggs in the fridge, and she didn't know that she needed to incubate them to take care of them as a parent in order to have success with her reproductive efforts. Um, all right, so nature shows love to show you examples of parental care, and as humans, we have very intensive parental care that lasts for a very long time. My last thought to leave you with today is thinking about asexual versus sexual reproduction, and what are the benefits of each one, and what are the costs? Why would it be good? Why would it be bad? And some of the questions to ask yourself to evaluate these are: how much genetic diversity do we find in the offspring, and is that a good or a bad thing? How far can the offspring disperse away from the site where they were born? So dispersal is moving away from, all right? So if you imagine a big tree and it's dropping its seeds, do they drop right down around it, or are they going to be transported away, right? So do they get dispersed? If we have a very small offspring, it will be more easily dispersed than if we have a much larger one. If we have an organism that does locomotion, it will be easier for offspring to disperse as well. If we do asexual reproduction, are we having few or many offspring? If we're doing sexual reproduction, are we having few or many offspring? If the environment is constant, or if the environment is changing, what type of reproduction would be better? And how much effort does it take for parents to do sexual reproduction compared to doing asexual reproduction? What are some circumstances that might be um, uh, risky or dangerous for parents if they are engaging in sexual reproduction or in asexual reproduction, right? So what are the costs? So we'll talk about these ideas when we meet in person, but those are the kinds of things that we want you to start developing an ability to think about and evaluate. But if you're not sure what the answers are to these, we'll go over them when we meet in person in class. All right, hopefully you have lots more questions about reproduction and how it works, and all the variety of different ways that organisms go about doing it. And we'll talk about those. I'll answer those questions when we meet in person.